Good morning and good afternoon to all of you for joining our live webinar today, Understanding Drug Efflux, Small Multidrug Resistance Family, Stoichiometry and Specificity. Today's webinar marks the first of 5 upcoming talks that will make up a part of our 2021 Research Insights Transporter webinar series. My name is Jason Villa-Gomez, Marketing Manager at Nanian Technologies, and will be your moderator for today's event. Before we get started, my colleague, Dr. Maria Barthelmess, Senior Scientist and Product Manager of the SURFER team, would like to say a few words.
Hello, everybody, and a warm welcome to everyone also from my side and on behalf of my colleagues, Cecilia and Andre. As Jason already mentioned, this webinar is the first one of a whole webinar series focusing on membrane transporters and SSM-based electrophysiology. We have scheduled 5 sessions with 10 scientists over the year. And we have never done a whole scientific seminar webinar series, but this year is somehow different. Due to the pandemic, we all are missing to visit conferences, listen to talks, and exchange experiences. We hope that with this webinar, we can contribute a little bit to keeping the community alive. We are thankful that so many people with really interesting projects agreed to present here. And I encourage you to contribute to a successful session by engaging in the Q&A after the talks. If you like to follow the series, please find more information on our website, and we also provide a link in the chat. The registration for the next session will start tomorrow. Actually, both of our speakers today won a SURFER instrumentation grant 2 years ago. We got a lot of requests to repeat this, and we had another one in last year. So maybe this becomes a tradition now, because I'm happy to announce that starting today, we are calling for proposals again. We are awarding a little instrumentation grant, including a SURFER instrument, consumables, and scientific support for half a year for an exciting transporter research project. So if you're interested, please find details on our webpage, and again, we will post the link in the chat. We are looking forward to receiving your proposals. So with that, I hand back to Jason to guide you through our webinar.
Thank you, Maria, for the exciting updates. We're definitely all looking forward to the upcoming SURFER grant. So joining us as presenters today is Dr. Randy Stockbridge, Assistant Professor, University of Michigan, and Dr. Katherine Henzler-Wildman, Assistant Professor, University of Wisconsin-Madison. Randy will provide a look at determining transport stoichiometry using SSME, so that's solid supported membrane electrophysiology, while Katherine will then proceed with the presentation, Basics of Promiscuity in Small Multidrug Resistant Transporters. Just a little bit of due diligence ahead of the talk. So we're going to have a live Q&A at the end of the presentation. So we both ask you to submit your questions in the chat window throughout the course of the talk. And if you have any technical difficulties at any point in time, feel free to write into the chat. That will come directly to me as well. It is to be noted that we will make an on-demand copy of this webinar available shortly upon completion. So do look out for that. And with that, I then turn over now to Randy for the start of the presentation.
Okay, thank you, Jason. Can you all hear me and see my slides right now? Yes, okay, excellent. I'm going to be talking about the promiscuity in this family today. Both Katie and I are going to be talking about the SMR transporters, so I thought I'd give a little bit of an introduction to the family as a whole as well. This is the small multidrug resistance, or SMR transporters. There we go. And these are widespread in the environment, and they're sort of most impactful. They're being spread around bacteria in environmental samples, continental scale pollution of estuaries, with these SMR proteins. And about 100 years ago, once humans first started applying quaternary ammonium antiseptics in hospital settings, these proteins first linked up with one of these mobile antibiotic resistance elements, the class 1 integron. And so the very first of this clinically important class 1 integron included an SMR protein and only an SMR protein. And these are still very widespread among antibiotic resistance gene cassettes. And by providing this fitness advantage to bacteria against these very general ubiquitous agricultural chemicals, antiseptics, industrial waste, that kind of thing, they sort of spread among bacterial populations and carry the antibiotic resistance genes with them. And so I think it's a very important thing to understand what these proteins recognize, what they transport, what chemical classes, and that will give us, some sense, some foothold to combat the spread of these proteins in response to these pollutants. And so what I'm showing you here is a sequence similarity network of the SMR family. And so here, each of these points represents a individual SMR sequence, and the closer together 2 points are, the more closely related the sequences are. And you can see that this sequence similarity network sort of forms these clusters, and often this clustering behavior represents functionally related groups of proteins. And it's a pretty compact sequence similarity network, so a protein in this cluster and one in this cluster will have about 40% identity and about 65% similarity. And most of the proteins that have been characterized thus far fall in this cluster right here, and with the larger dots I'm showing you ones that have been biochemically characterized in some way. And these have been shown to transport these sort of polyaromatic cations, like I'm showing here. They're proton-coupled transporters, and among these proteins, the protein EMRE, which Katie will be talking about in a few minutes, is sort of the exemplar of the family. Very well studied, very well characterized. But you can see that there's all of this sequence space over here that had no assigned function as of a couple of years ago. People had shown that proteins from within this cluster were not capable of transporting molecules that look like these big polyaromatic cations, but nobody really knew what it was that they did. And so we sort of got interested in this upon the discovery of bacterial operons that actually handle guanidinium. So they're dedicated to guanidinium metabolism, transformation, or there's ribose switches. This is a crystal structure of a ribose switch. You can see a guanidinium ion bound right here that turn on genes. And what we noticed was that the proteins in these clusters were very frequently associated with these operons and these ribose switches. And so we characterized proteins from each of these major clusters and asked a very simple question, if these operons are dedicated toward guanidinium metabolism, maybe the exporters are guanidinium transport proteins. And so we kind of initiated this project with radioactive uptake experiments as our main tool. And we saw that we were able to obtain radioactive guanidinium, and we see very robust guanidinium uptake catalyzed by proteoliposomes containing the proteins from that other cluster. And we were able to sort of get our hands on other radioactive substrates, mostly biological type molecules. And we showed that, among these types of molecules, proteins from that other cluster were very selective for guanidinium and selective against these other kinds of biological metabolites. And so with that, we sort of re-annotated the SMR family and said, okay, the proteins in these clusters seem to be guanidinium exporters. And we continued to characterize them. Showed eventually that they had 2 proton to 1 guanidinium stoichiometry, so they were electrogenic. And interestingly, the proteins from this blue cluster over here did not transport guanidinium ion. They were selective against it. And so sort of the situation a couple of years ago was that there seemed to be this very binary split in the family where the proteins from the drug cluster, like EMRE, would transport canonical EMRE drugs, and the proteins from that new cluster, which we're calling the GDX cluster, did not. And meanwhile, EMRE would not transport guanidinium. The proteins from the GDX cluster would, and both were selective against sort of common biological metabolites with guanidinal groups. But this still wasn't, totally satisfying for us, because we still hadn't gotten at this question of the spread of the multi-drug resistance gene cassettes. And one thing that we noticed, in contrast to the riboswitch associated GDX proteins in yellow, which are, very strictly segregated toward the GDX cluster, both clusters are very frequently associated with these mobile resistance elements, either integrons or plasmids. And so it seemed like we didn't quite have the full story here. Guanidinium ion itself is not a common environmental pollutant. So why are we still seeing this frequent association of these proteins with horizontal gene transfer elements? And so to do this, we just had to expand our capabilities to measure transport by these proteins. There's just simply not enough radioactive substrates available to do what we want to do here. And so this is when we started using the SSM electrophysiology. And as I mentioned before, both subtypes are electrogenic proteins. And so what we're seeing for the GDX protein specifically is the import of 1 guanidinium coupled to the export of 2 protons. And so this is what the data looks like. And what you can see is that when we perfuse our proteoliposomes that are capacitively coupled to our measurement electrode with guanidinium, we see this nice peak here, capacitive current that is in the direction that we would expect for 1 guanidinium in, 2 protons out. That's a net negative. And then when we that quickly reaches steady state as equilibrium is obtained, we then perfuse with the non-activating solution, and we see a capacitive current with the opposite polarity representing the guanidinium that had accumulated in these liposomes now flowing out. And so we can do experiments like substrate titration. So this is a guanidinium titration. And we get nice Michaelis-Menten kinetics out of this. And so we were very excited now, because this sort of provided a general way of testing different substrates, and we could really do a substrate activity relationship now to see how you could tweak the substrate and whether you would get transport or not. What are the substrate rules for transports by exemplars of these 2 subtypes in the SMR family. And so collected a lot of data. This is sort of representative where we perfuse with empty liposomes, and we're pretty much baseline with all of these different substrates with these different chemical structures. And then we were able to test both one of our representative GDX proteins and our representative drug exporter EMRE. And you can see for some substrates like guanidinium are robustly transported by the GDX proteins and not by EMRE, for example. There are other examples that are transported by EMRE and not the GDX proteins. But there's also a great deal of substrate overlap, right? We're seeing pretty nice currents for many, many substrates. And so to just synthesize all this data now into kind of what are the rules for substrate transport by proteins in the family. So the GDX proteins, of course, we see nice guanidinium transport. We also see very, very robust transport of singly substituted guanidinium ions. So you can even put, a big phenyl group on one of the nitrogens of the guanidinium and still see very good substrate transport. And doubly substituted guanidiniums for this protein are transported pretty well also. So you can put a single methyl group on opposite nitrogens or in the same nitrogen and still see some degree of transport. Where things fall apart is sort of any additional substitution beyond that. So tetramethyl with 4 methyl groups is not transported at all. Ammonium type compounds are not transported. And then, of course, the sort of biological compounds guanidinyl compounds like arginine and agmatine that we mentioned, we also see no currents for that. We characterized a few other of the GDX proteins and found that this sort of substrate promiscuity is common traits in the GDX section of the family where we are seeing this robust transport of, for example, phenylguanidinium. Okay, and so now if we compare the substrate transport for EMRE, what we see at the outset is that, as we mentioned before, guanidinium is not transported. But as soon as you start substituting this guanidinium ion with hydrophobic substituents, methyl, ethyl, especially phenylguanidinium, you start to see much more robust currents. It's transporting very capably. It is fine with double substitution. EMRE is fine with a tetramethyl guanidinium. This quaternary ammonium, tetramethyl ammonium, is kind of transported most robustly out of all of these. And once again, we're retaining this sort of strict selectivity against biological metabolites like arginine and agmatine, which makes a lot of sense. You don't want these proteins to be exporting your valuable metabolites. And then another thing that we see transported by EMRE is metformin, which is a common diabetes drug that is in water supplies and water treatment plants and so forth. And so, the picture that we're seeing emerge is not, what we thought initially, that there was this big leap in specificity. The guanidinium exporters transport guanidinium, the drug exporters transport these polyaromatics, and there's little overlap between them. In fact, what we've seen happen is that the drug exporters have gained some kind of requirement for more bulk and hydrophobicity in their substrates. They've lost a requirement for the number of H bonds. So for example, the tetramethyl will be transported fine. But overall, there's a lot of overlap in the family too. And so we had all of this data in hand when we finally solved the A structure of one of these proteins. And this is a GDX protein, GDX-CLO. This protein is an anti-parallel dimer. So in fact, the open state and the outward open and inward open state are, essentially identical, just related by twofold symmetry. And so we can put together kind of this view of of how this, transport would, or the outward to inward open would look like. And what you can see from this structure, the two subunits of the dimer, we have this wide aqueous vestibule that goes right down to these glutamate residues that bind the positively charged substrate. And then when there's this conformational exchange, now we've got the aqueous vestibule open to the other side. And this is a view we crystallized with phenylguanidinium in the binding site. That was convenient because it was large enough to be really clear crystallographically. And so here's the phenylguanidinium, and you can see it sort of poised right between the two glutamates here and here. But I don't want to go into great detail about the details of the binding site. I want to focus on another feature of this protein, which I think connects nicely back to the SSM electrophysiology experiments we did. And that's what we call the membrane portal. So if we look at this sort of surface representation of our GDX protein, the approximate boundaries of the membrane are indicated by this yellow box here. And we see that two of the helices sort of splay apart so that there's almost like this gap, this dip down into the membrane. And it seemed like kind of an unusual feature, especially I think these types of portals have been seen in things that, to provide access to a binding site for something very hydrophobic that maybe partitions into the membrane. But guanidinium is, ultra soluble and is, it's not clear why that access would be required. And so to sort of interrogate this a little bit more, we decided to solve the structure with this compound here, kind of bring this hydrophobic substituted guanidinium to the most extreme extent. And this is octyl guanidinium. And what you can see in this octyl guanidinium bound structure, you're actually looking like right down the tail of the octyl guanidinium with the head group pointing away from you and the tail pointing at you. And you can see that the head group binds between these glutamates, and this octyl tail is able to stick out into the membrane. And this is kind of a top view of this. This is a slice through the center of the membrane, and you can see the octyl interacting with the the guanidinium interacting with the glutamates, while the octyl tail sticks out into the membrane. And so this, I think, kind of connects to our SSM electrophysiology experiments, where we were seeing that signal substituents on the guanidinium were still transported very well. And at the same time, something like arginine and agmatine were not. And I've indicated with an arrow right here where sort of the polar amino group would be on agmatine, or amino plus carboxylate would be on arginine. And you could see that that would be very unfavorable to have that sort of facing the membrane. And so we think this portal might actually be a conserved feature to select against things like arginine and agmatine, prevent that from binding. And this is sort of what the evolutionary people like to call cryptic transport function. There's sort of been high evolutionary pressure to exclude agmatine, metabolite from these proteins. But, throughout evolution, these proteins haven't known or cared about things like phenylguanidinium or ethylguanidinium. And so there is no evolutionary pressure one way or the other to exclude these. And so these features just happen to be there in these proteins. And once we as humanity started putting things in the environment like diphenylguanidinium from the vulcanization of rubber, or metformin, that diabetes drug I mentioned, agricultural chemicals like dodecylguanidinium and various quaternary ammoniums, these proteins were able to sort of step into that role very naturally because they had this feature that happened to be conserved. And so that is the story there in a nutshell, and thanks for your attention. And I guess Katie's going to go now, and I'll be happy to take questions afterwards.
Perfect. Thank you, Randy, for going and walking us through your presentation. Katie, I now open the floor up to you for your presentation. And again, I'd just like to remind everyone, feel free to send in your questions directly into the chat. That comes to me, that goes into the queue, and then we'll address them individually for Randy and Kathy at the end of the talk.
Okay, thank you. So today I'm going to talk about what we've done to use solid-supported membrane electrophysiology to look at the transport stoichiometry. So this is a slightly different question than what Randy was addressing. She was looking at the substrate promiscuity of transporters in this family. And we're looking more at mechanistic promiscuity. So our NMR experiments and other biophysical studies suggest that although this 2 proton for 1 substrate antiport pathway that I've shown in orange is the dominant transport mode for the known substrates of EMRE, these other pathways may be possible, particularly if you're transporting some of these other substrates or under different environmental conditions. And so we wanted to look at whether EMRE is really a tightly coupled 2-to-1 antiporter, or if the stoichiometry varies with these different conditions. But first we needed an assay that was quantitative and would let us look at this question for all these different substrates and different pHs without using NMR quantities of transporter for every single assay that we ran, which some of our previous transport assays have used. So we were really looking for an assay to look at this stoichiometry question that would work for this very diverse set of substrates. And Randy's already explained how the SSM electrophysiology really helps because you don't require these radio-labeled or fluorescent indicators. In particular for EMRE, a lot of the substrates are fluorescent or they're polyaromatic cations. And so trying to use some sort of fluorescent pH readout becomes extremely difficult because of interaction between the substrate and your pH sensitive fluorophore. We need something that we can adapt to different pH conditions, and we want something that can be done efficiently because we want to screen a really wide variety of conditions for the EMRE assays. Okay. Now, I'm not going to actually talk about EMRE much in the rest of this talk. I'm going to switch gears because if we're going to develop an assay to look at stoichiometry using SSM electrophysiology, then we need known, well-characterized systems. So I'm actually going to start with GDX, which, as Randy just told you, is this is written backwards, 2 proton to 1 guanidinium transporter. And as you can see, SSM electrophysiology has previously been used to monitor the transport, so it was a great starting system for us. So this is the previous characterization of the transport stoichiometry that was done by Randy's group. So this is a very standard reversal potential assay where you're varying the potential that is set across the membrane, and then using a pH-sensitive dye to watch the movement of proton in response to that potential as the system reaches equilibrium. And so in this case, you can set different membrane potentials and look for the condition where you are at equilibrium with your concentration gradients and your membrane potential. So you can plot this accumulated change in substrate, find the 0 point where you're at equilibrium, and then calculate the stoichiometry of 2 proton for 1 guanidinium. So here is how we're going to set up an assay to go after the same idea, but in a slightly different format using the solid supported membrane electrophysiology. So we're going to start with our initial buffer conditions where we have no gradient for either proton or guanidinium. And then we're going to flow in our activating buffer, and this is going to set up a 2-fold proton gradient, but again, no gradient in guanidinium. So this creates a 2-fold outward proton gradient that's going to drive net transport, and as Randy showed, we get this nice negative current, exactly as you'd expect. And as we return to our initial buffer, you can see the reversal of that process. And then what we can do is we can change our activating buffer so that we don't just create a 2-fold proton gradient, but we create a variety of different guanidinium gradients as well. And here you'll see the guanidinium gradient is large enough and in the opposing direction that it drives net transport in the opposite way pushing protons back in against this twofold gradient. And you can see that now we have a positive current rather than a negative current. So what we can do in this assay is vary those guanidinium ratios. So this is showing you the ratio of guanidinium, the gradient across the membrane that we've created, and look for this reversal condition where there's no net flux, indicating that we're at equilibrium. So this is a constant twofold proton gradient and variable guanidinium gradient across the different conditions. And I've shown a couple of traces here. These are repeats on the same sensor, and you can see that they're virtually indistinguishable. And I'll talk more about different sensors in a minute. But stoichiometry is a thermodynamic property. So what we're really interested in is the net charge movement to reach equilibrium. Here's just some of the math. When we set up this initial condition, we do not have a voltage. We only have concentration gradients. And if we're at the reversal condition where there's no net flux, then the free energy is 0. And so at that point, you can very easily calculate the transport stoichiometry from the ratio of the concentration gradients that you use to set up the assay. So looking back at our data, like I said, we're not so interested in the kinetics here. We're interested in how much charge movement there is in total to reach equilibrium. And so we can integrate these traces. And if we integrate the currents, now you can see that we reach a very nice equilibrium state where we're watching just the accumulation of net charge that's moved across the membrane under these different conditions. And you can see very nicely that we have a reversal condition here at a 4:1 guanidinium ratio. And then we can plot this net charge that's moved to reach equilibrium as a function of the ratio of the concentration gradients. And we're writing it in shorthand this way, but this is really what is shown on this axis. And then this crossing point then is exactly your stoichiometry. So where it crosses 0 is the transport stoichiometry. And here I'm showing this just compared to a traditional reversal potential assay so you can see the analogy. So in a reversal potential, you're setting different transmembrane voltages and watching the accumulated change in substrate concentration or proton concentration as you reach equilibrium, looking for the point where it crosses 0, which is shown here when the data is plotted as a function of the transmembrane voltage. And here we're setting different concentration gradients, and now we're monitoring the accumulated charge movement to reach equilibrium under each of these conditions, looking for the reversal condition, which is where it would cross 0, and reading off the transport stoichiometry. So let's look at some of the controls and things you might think about in evaluating the assay. So this is the current that we get for 4 different sensors running exactly the same assay. You can see that it's very reproducible. There is some difference in the peak current from sensor to sensor, and this is primarily because you have a different number of liposomes that actually adhere on the surface of each sensor. And so we can integrate the current the way I showed previously, and you can see there's a little bit of variation here, but importantly, there is no variation in the point where they cross 0. So the total net charge movement depends on how many proteoliposomes you're recording that capacitative current from. But the 0 crossing point where you're at equilibrium, what concentration gradients are at equilibrium, does not change. And so we can take the total integrated current for a given sensor, and we can use that to normalize the data, basically saying we have a lot more current on this sensor, less current on that sensor to account for the number of proteoliposomes. And then when you normalize that way, you can see that the data is very, very consistent across the different sensors. And in these integrated current traces that I've shown you, you can see the dark line, that's the average, and then the shaded area represents the standard error at each point along the trace. So the error in the data is really quite small. We can also vary the lipid to protein ratio. And so if you have more or less GDX incorporated into the proteoliposomes, that changes the kinetics and the peak current for this transport process. But if you integrate the total current, that doesn't really change. So this is shown on the same scale, and your reversal condition again does not change. That's exactly as you'd expect, again, because we're looking at the transport stoichiometry, which is a thermodynamic property. We can also subtract the background for any sort of non-specific interaction of the ions and the substrates with the liposomes or leak through the liposomes non-specifically. And so we can run control experiments, either with a non-functional point mutant of GDX incorporated in the liposomes, or we can use empty liposomes. And in each case, the currents are quite small, and the total net charge movement as we integrate the currents is very slow. We can subtract either of those baselines, again, from either of the lipid to protein ratio experiments, and they give you a stoichiometry of 2 to 1, 2 protons for 1 guanidinium with an error in each case. We can also change the internal solution. And so this is really useful because if you want to screen a whole bunch of different conditions, traditionally you have to make a separate batch of liposomes with each internal solution condition. So we can do extensive washes with a different condition to change the internal solution. And so here we've changed the internal proton concentration from 100 nanomolar to 200 nanomolar with the liposomes already adhered on the sensor. And we can repeat this assay, and you can see that again, you get reversal at exactly the condition you would expect for 2 protons to 1 guanidinium antiport. And this is important because this confirms the fact that we get exactly 2 to 1, again, confirms that we have switched this internal proton concentration, and it is exactly what we think it is. If it was still 100 nanomolar, then this would not cross in the correct place. More interestingly, we can also switch the internal guanidinium concentration. So here I'm showing you data for 500 micromolar internal guanidinium and 1 millimolar internal guanidinium. These were run on the same sensor, and again, you get 2 to 1 antiport with an error very precisely. So that's useful because now we can change the internal conditions and run a whole bunch of different assays to really probe how the relative concentrations and gradients affect the transport stoichiometry. But to establish an assay, we need to look at more than one system. And the second system we looked at is a CLC, EC1. There's a couple of reasons we chose this. One is that we know that it has fast transport. You've got good currents for the SSM electrophysiology readout. But it also has this property that it does 2 to 1 coupled antiport of chloride, but a much more loosely coupled transport of nitrate. So this will be a good test of, can our assay distinguish these different kinds of behavior? So we're going to set the assay up very similarly, where we'll set up a 2-fold proton gradient across the liposomes with our activating buffer. And then with the activating buffer, we'll set various chloride gradients, either no chloride gradient or 60 to 150 millimolar outward chloride gradient. So these are all outward gradients. And again, you can see that we get reversal as we'd expect for a coupled transport. The difference here is that with chloride, we do have a significant signal with our empty liposome current traces. So there is a background signal from the chloride interacting with the liposomes and potentially leaking through the liposomes. And I'll talk about how we account for that in a minute. We can also run it backwards, where we have the proton gradient inward and the chloride gradient inward. And again, we can get reversal. The kinetics are different. The empty liposome current traces look a little different. But when we integrate the data, so this is the raw data for proteoliposomes with CLC EC1. This is the gradients in the same direction, but looking just at empty liposomes, so that'd be the background signal due to the chloride interaction and leak. And when we subtract out the background from each of these, whether we have the inwardly or outwardly directed gradients, you can see that both of them cross at exactly the ratio you would expect for 2 chloride to 1 proton. So remember, this is the way we're setting this up. 2 to 1 would be 2 protons for 1 chloride, 0.5 is 2 chlorides to 1 proton. So that's exactly repeating the known stoichiometry for chloride for CLC. So what about this background signal? So here's the integrated current that you see for the empty liposomes. You can see that it is a significant amount of net charge over the course of a second. And there's a fair amount of variation here between different sensors. And if you look just at the raw current traces for the proteoliposomes with CLC, you can see that they reach equilibrium very nicely. It's a very steady state if you don't have a chloride gradient, but with an increasing chloride gradient, there's more of this drift over time due to the chloride interaction and leak through the liposomes. So, however, when we subtract the background signal from the empty liposomes, now the data looks very nice again. So even though we have this significant background signal from the chloride interacting non-specifically with the liposomes rather than with the transporter, we can subtract that out effectively and get the reversal condition very cleanly. You'll notice that the error bars are a little bit larger here than they are here. There's a couple differences. We had a larger proton gradient here, so we're going to drive a larger transport current, coupled transport current, and the total chloride concentration is smaller here. So I think that if you have an ion that's going to interact with your membranes or potentially leak through the membranes, you can minimize the error in these assays by manipulating your conditions to enhance transport relative to leak and minimizing the concentration of the ion that's causing problems. Last, I want to talk about nitrate. So this should be more weakly coupled. So again, we're going to run the assay two ways, one with inward facing gradients, one with outward facing gradients. Again, we get reversal of the current as we vary the relative gradient of nitrate compared to our proton gradient. And again, we have very little signal in the empty liposome current traces. So there's not a large background for nitrate. And now when we subtract out the background and we look at the reversal condition, it's right about a 4 nitrate to 1 proton stoichiometry. So this is the apparent transport stoichiometry, but is this really coupled, strictly coupled 4 nitrate per 1 proton? Or is this a combination of some sort of coupled antiport, perhaps 2 nitrate to 1 proton like you see for chloride, plus additional nitrate leak? So we tried to get at this by doing the assay a little bit differently. So here we've set up a constant proton gradient and we have no anion gradient. So equal concentrations of anion on both sides. So proton is going to drive the coupled transport. And then we're going to vary the total anion concentration. So if you have a tightly coupled transport process, all that should matter is the relative gradients of the two ions, and that's constant under all these conditions. The exact concentration of either ion shouldn't really matter that much. However, if you have a mix of leak plus coupled transport, then the relative concentrations of the two ions matter because that's going to shift the balance or the partitioning between leak and transport pathways. If you have a lot more nitrate than proton, that's going to favor more leak. And if you have less nitrate, then transport is going to be a greater proportion of your signal. So we ran this assay with different concentrations of chloride. Remember, 2 to 1 chloride is we thought to be a tightly coupled strict stoichiometry. And we can see that there's no difference in the traces when you vary this chloride concentration. So that's as we'd expect for tightly coupled antiport. However, when we look at nitrate, now we do see a significant change depending on the nitrate concentration. And if you integrate this and plot the total net charge to reach equilibrium, now that you can see that it's highly dependent on the anion concentration. So changing nitrate does affect the current, and this indicates that you likely have a combination of coupled antiport of nitrate and proton plus an additional nitrate leak. So to summarize, so far this assay has worked quantitatively to recapitulate the known transport stoichiometries of both CLC EC1 and GDX. The empty liposomes seem to provide a pretty good control for background, and we can quantitatively exchange the internal buffer, which is a real advantage when you want to screen a lot of different conditions. We can also determine the transport stoichiometry even when you have an ion that is going to absorb or leak through your membranes and give you a large background signal. And I think we can start to distinguish these ideas of, do we have a different transport stoichiometry or do we have transport plus coupled transport plus leak pathways? We really like this assay because it does not use a lot of transporter, and we can really quickly screen a lot of different substrates and pH conditions. And then just to thank people who provided us with plasmids or protein to get started on this. Nathan Thomas is the student in the group who did most of the work. And I'm happy to take any questions.
Thank you, Katherine. I'll go ahead now and share my screen again, and I'll start moderating the questions that we received so far. And while I get this set up, I also welcome you all to continue to write the questions. They will be added directly to myself here. So I'll just go through and work through Randy. Received a very welcome compliment, beautiful work. And to paraphrase a little bit, I was wondering if any of the GDX are heterodimers, or are you working on the assumption they are homodimers? That was for Randy.
Yeah, thanks for that question. So the ones that we are, that I spoke mostly about are homodimers. So you may remember in that sequence similarity network I showed, there are actually 3 major nodes in the GDX cluster. The biggest node were homodimers, and then the other 2 smaller nodes were actually an inward facing and an outward facing subunit of a heterodimer. And we have biochemically characterized, excuse me, 1 of the heterodimer, 2 of the heterodimers, I think, just, you know, the very sort of surface level characterization, showing that they transport guanidinium and that they behave as a heterodimer, that you need both subunits there.
Thank you. And then as another question, did you see major differences in current trace kinetics across the different substrates? For example, faster decay of the signals. Could you identify substrates with electrogenically bind to the transporter but are not transported?
Right, that's a good question as well. So, I'll answer the binding question first. You know, I think just with the positively charged substrate, if we were to get sort of a binding event but not a transport event, we would see a positive current rather than the negative current. You really need the 2 protons going the opposite direction in order to get that negative current. So that's a nice thing about this system that makes us feel a little bit more confident that we're looking at binding and not, I mean, that we're looking at transport and not binding. And then let's see, what was the other part of that question?
So I'll just go from the top. Did you see major differences in current trace kinetics across the different substrates? For example, faster decay of the signals. Could you identify substrates which electrogenically bind to the transporter but are not transported?
Mm-hmm. Yeah, these all do look sort of like transporter-ish kinetics to us. It's not the very, very sharp current that people have seen, for example, with ion binding to an ion channel. But for sure, we see differences in the kinetics. I mean, I think that would be expected, that these various substrates are transported at different rates, and that you know, the decay, which sort of represents the rate at which this charges, is, you know, capacitively charging the liposome, you know, that's different from substrate to substrate. We haven't analyzed that in detail at this point yet, although that may be the next sort of frontier for us.
And then I guess this one can be addressed possibly to you both. Can you go over washing to change internal solutions? Can you change any internal solution? So maybe you can each comment a little bit on your experiences.
We've done it with changing the internal proton concentration and guanidinium concentration and chloride concentration for CLC. I didn't show that data, but we haven't looked at some of the other, like we haven't done it with bulky EMRE substrates. So I think there's a limit to the number of experiments we've done so far.
Yeah, I mean, you can see the exchange with pH as in my experience, and I really haven't tried it with much else. But pH is, I think, rather easy to equilibrate.
And then now for Katherine, could you explain again why a tightly coupled transporter in CLC means there is no change in signal when you change the anion concentration? Thermodynamic considerations would say that higher anion gradient would still change the integrated current.
Yeah, we're not changing it hugely, right? We're only changing it a few fold. And if you have, so what we're looking for is a reversal condition. And then here where we're running not at a reversal condition, so we actually get a current. As we change that gradient, you're not really like the proton is driving chloride transport, but there's no chloride gradient. So we're running it under a very particular condition, right? So I think what's important to remember is we're starting with equal chloride on both sides and a constant proton gradient, and then we're running it with either, 5 millimolar or 10 millimolar, 30 millimolar chloride on both sides. So there's no chloride gradient. So in the absence of a gradient of chloride, then if it's tightly coupled, you shouldn't see a change in that integrated current, because the only thing driving the transport is the proton gradient.
And then maybe you can each comment on this as well. This is more, how does overuse of a sensor manifest? Noisier data or something else?
I guess I'm not sure what overuse would mean. I mean, at some point we just get it very noisy and we start to see sort of rapid degradation of the currents we're seeing. So, I think when we do these experiments, what we always do is kind of a positive control perfusion at the beginning where we perfuse with guanidinium. We say, okay, we see a nice current there. Then we perfuse with our test substrate, say it's dimethyl guanidinium. Usually we see a smaller current. And then we go back to the original guanidinium, perfuse with that, and we say, okay, is this first guanidinium current the same size as the, our third trace, our second guanidinium collection? And if that's not happening, which, that happens eventually with these sensors, then we just throw out that last little bit of data. So we kind of go back to this positive control throughout the course of using one of these sensors.
Yeah, we also see that the data gets very noisy. You can run a lot of experiments on one sensor, but eventually it does start to degrade and the data gets noisier. And we also tend to run the same experiment, like what your initial experiment, you run again periodically to just check that you're still getting consistent data.
And then for Randy, perhaps someone that joined a little bit late, maybe I missed it, but for the SSM assay with the substrate that has a lipid tail, did you add this to the liposomes or to the activation buffer?
Let's see. So the octoguanidinium, the one with the tail, we didn't, it's a detergent and it destabilizes the liposomes and it doesn't work. So we haven't tried that with transport. With the other substrates, we can go the diphenyl, the tetrapropyl ammonium we've done. All of these we just add to the perfusion buffer. And, there's a point eventually with higher concentrations of these more hydrophobic things where you start to see with the empty liposome control kind of a intolerable amount of kind of non-specific interaction. And so because these are positively charged substrates, this manifests as a positive current in the empty liposome control. So there is sort of a point that's related to the hydrophobicity and the concentration where we can't really do the experiment and trust it. But I don't know. I feel like we're also going pretty hydrophobic and still getting reasonable results and still not getting this high background current. And, and again, it's an easy control to do it with the empty liposomes and check that.
Then while you were commenting on control, what is the ideal control experiment? Empty liposomes or proteoliposomes with non-functional transport? I guess maybe you can each comment as well on your experiences here.
Yeah, that one, from our experiments, they both seem to work really well. So one of the things we think about goes back to one of the earlier questions. Is there, say, an additional binding site where you could get binding and not transport that you could potentially get with a non-functional mutant like this E13Q GDX? And so we worry about getting some current due to binding that's not due to transport in that situation. Obviously, we're not empirically, we are not seeing that here. But I think in that scenario, if you're worried about that, then an empty liposome actually makes a better control system.
Yeah, I mean, it depends on what you're controlling for. If it's just the substrates partitioning into the membrane, then the empty liposomes are fine for that. Yeah, I mean, as with everything, I think it's worth trying everything. And, you know, there's no answer to what the ideal control is. You know, if you've got something that's locked or that, you know, doesn't bind substrate, then that might be a fine control to tease out whether you're seeing binding currents or not. You know, it just depends on the system, I think.
Yeah. And then for Katie, will this assay work to determine the stoichiometry of any transporter? What are the limits of the assay?
So we've gone, I think, I'm going to interpret this in terms of like transport rates. So obviously, the faster your transport is, then the higher your current. And so the easier it is to read out with the SSM electrophysiology. So faster transporters are better. That said, we've gone as slow. We've done some assays with EMRE where we know from the NMR experiments what the alternating access rates are. And so that sets an upper limit for the rate of transport. And we've gone as slow as 0.5 per second and still detected reversible transport currents. But I would say you probably can't go a lot slower than that.
Perfect. Well, for my part, I want to thank you both for your presentations and the contributions to the first webinar of the series. I leave the floor open for a couple more minutes for any last questions that might come in case we're still a little bit under the hour. So we're still here with the presenters if there are any open questions. In the meantime, I just shift between the upcoming events so that you each can have a little bit of notice, and as well as the last reminder that now we have opened up the call for the Surfer N1 grant. So we look forward to your exciting proposals on that. And as always, this will be reproduced on demand. So if there are any questions that come to mind after the fact, feel free to get in touch with us. We will then naturally pass that along to the presenters, and we will make a transcribed copy of the Q&A also available. So please don't hesitate to rewatch this as well. Okay, perfect. So then I leave it at that. Maria, if you would like to say any closing words, then we would go ahead and let everyone get on to a nice rest of their day.
Yeah, just thanks to everybody. I really enjoyed that, Randy and Katie. Thanks a lot for presenting here.
Thank you.
Thank you.
Thank you so much. We wish everyone a great rest of your day.